The recently published paper is about a potential drug candidate that we have identified based on a number of years of research here that was identified as a potential agent for Alzheimer's disease and other diseases where neurofibrillary tangles are formed, which are these deposits that are found in the Alzheimer's brain composed of the protein tau, and in particular misfolded forms of the protein tau. And so we believe that one of the consequences of neurofibrillary tangle formation in Alzheimer's and other diseases like frontotemporal dementias, for example, is that the tau, which has a normal role in the cell, when it becomes deposited in these aggregates, loses its ability to do its normal job. And that normal job is to essentially stabilize structures known as microtubules, which by analogy can be thought of as the railroad tracks within the cells on which various cellular cargo is shuttled. And one of the major consequences of these tau depositions is a disruption of these railroad tracks or these microtubules, such that cellular cargo is not properly shuttled up and down the cell. That leads to neuron damage, neurons being the cells that, of course, are involved in learning and memory, behavior, and hence you have the neurodegeneration ultimately that you see in these diseases. And so the thought was, could we potentially find drugs that would stabilize these microtubules, these railroad tracks that appear to be disrupted in these diseases due to the loss of tau function? And so this paper is about a drug that we have characterized called epothilone D that was previously utilized for cancer treatment in clinical testing, progressed to phase 2 clinical testing in cancer patients, and have shown that this drug, which gets into the brain quite nicely, does seem to, in fact, stabilize these microtubules and works in our animal models of Alzheimer's disease and tau deposition. And so we saw an increased microtubule density in the axons and neurons of these nerve cells within these transgenic mice over those that didn't receive the drug. That resulted in improved axon function. We can measure things known as fast axonal transport. We saw increases in axonal transport, and that seemed to result then in an increased survival of the neurons themselves. So there's a region in the brain called the hippocampus where these animals show a lot of neuron loss. We prevented much of that, and somewhat surprisingly to us, we also prevented further deposition of tau into insoluble inclusions. We didn't necessarily anticipate that because this drug was designed just to compensate for the tau loss of function, not to block its, the misfolding of the tau that's there. But it seemed to do that, and it's a little bit complicated why that might be. We don't have a full answer, but we were certainly pleased to see that that was the case. So the doses we used in our animal studies were about 1/30th to 1/100th the level that was used in human equivalent doses in the cancer patients. And so it's possible this drug will be much safer at those doses in humans. And so Bristol Myers Squibb is in the process now of testing these lower doses in Alzheimer's patients, see one if it's safely tolerated. And then they'll also look at things like CSF levels of tau to see if they're having any impact on cerebrospinal fluid tau levels, which is a biomarker of Alzheimer's disease. They'll also do some cognitive testing in this study, although it's a small study, so they won't get significant results, but they might get a hint of something happening. So the drug is moving forward into clinical testing.